We spent the first part of our course learning about cells and living things, and microbes in particular, how they're classified and their growth requirements. Now we turn our attention to how to control them, first outside the body using physical and chemical methods, and then inside the body using vaccines, our immune system, and antimicrobial drugs. One consideration you'll learn about in the videos I made is what type of microbe are we trying to kill? If it's a bacterial spore, those are really resistant to drying, to heat, and to some chemicals. There's a fun lab experiment posted for you in which someone tries to do exactly that, kill a bacterial endospore. Our notable scientists for this week were all involved in environmental microbial control since they were working to prevent disease in a hospital setting. Florence Nightingale, Joseph Lister, and Ignaz Semmelweis. You might recognize one of the voices in the Semmelweis talk. That's Carl Zimmer, the science reporter who did the extra credit presentation on viruses that some of you were able to watch. Your enrichment continues to explore germs and how to kill them. It's an interesting phenomenon of human nature that we tend to think of some places like the toilet bowl as being extremely germy, but things we handle all the time, like the TV remote or our cell phones, are considered safe. These videos might change your perceptions a little bit. We're going to learn about how bleach works to disable microbes, and you have a little worksheet to do as you listen to that podcast. I want to give you the opportunity to research a microbial control method of your own and post it in the discussion board. I'm hoping that some of you will have unique experiences to tell us about, like pasteurization or antisepsis prior to performing a C-section, disinfecting a hospital room or an ambulance after a patient has occupied it. These controls are not limited to hospital settings. If you work in a restaurant or a nail salon or even the public library, any public place, there's a higher standard of cleaning expected when the public is served, and we'd love to hear about it. Lastly, we do our second Teach the Class quiz, this time on respiratory infections. Same pattern as before. Review the slide presentation that you and your classmates made. Print out the presentation slides using the printable format to save yourself some paper and ink, and use this as a resource for the quiz. The format is the same, 15 multiple choice questions in 20 minutes. Once we're done with this week, we have finished 1/3 of the course. I'll give you the opportunity to evaluate how it's going in a survey so that we can make adjustments going forward. After this week, I will also have a pretty good idea of how many points we will finish the course with based on what we've done so far. This is going to be a busy week, but also lots of fun topics, really interactive. I think you're going to really like it.